Everyone. Thank you so much for this kind invitation. My name is Jackie Bliley, and I'm currently a postdoctoral fellow in Adam Feinberg's lab at Carnegie Mellon University. And today I'm going to be talking about some of the work we've been doing with 3D bioprinting with a specific focus on the human heart. So now to move on to the meat of my talk, let's talk about the motivation for building these heart muscle tissues, which is really that the heart has this limited capacity to repair itself following significant damage or disease. This can sometimes result in dramatic functional and structural remodeling of the heart muscle, as you can see in the example case of heart failure on the right. With really the main outcome in both of these examples being that the heart's not able to pump enough blood to meet the body's demands. The only long-term treatment for heart failure is a heart transplant. But unfortunately, if we look at a graph of your survival probability as a number of function days on the wait list, I want to draw your attention to this red line, which these are the most severe patients on the wait list. And you see that after about 1 year of survival, these patients have about a 48% chance of survival after waiting 1 year on the wait list. I'm sorry. And it's important to keep in mind that these are the patients who actually qualify for the wait list, with many patients being excluded. Thus, there really is this need to develop therapeutic alternatives that are capable of repairing or replacing the human heart. Engineered heart muscle tissues have emerged as this in vitro tool or even as an in vivo therapeutic option where you can obtain these patient-specific somatic cells. These cells can then be reprogrammed to become like cells during embryonic heart development or induced pluripotent stem cells, and then can come on to become many different tissues and organ types present within your body, including heart muscle cells or cardiomyocytes. These patient-specific cells can be used to make engineered heart muscles or used to study drug toxicity or model disease progression in a patient-specific manner, or even as an in vivo therapy to repair the damaged heart. However, one key challenge we face if we're looking to create a therapeutic option is that these current fabrication approaches are unable to build a fully functional human heart for replacement. So what kind of biofabrication approaches are needed to build a human heart? And this probably relies on what you're trying to make, but if we're thinking about the adult human heart, we know we have over 10 major extracellular matrix proteins, which are very abundant in the heart. We have over 11 cell types, varying elastic moduli, and a multiscale vascular network ranging from 5 microns to up to 5 millimeters in diameter. Thus, we really need a biofabrication approach that can spatially control the composition, the microstructure, the macrostructure, the mechanics, and cellularity to make these tissues and organs. And 3D printing is one potential approach to build these heart muscle and tissue and organs for repair and replacement. And you all are probably familiar with traditional 3D printing or fused deposition modeling, which really relies on the softening of thermoplastic material, which is shown in orange here, through a heated extruder. This can then be deposited layer by layer, as you can see on the left, to create a 3D structure. However, because these thermoplastics are much more rigid when compared to the soft materials like cells and extracellular matrix that makes up tissues in our body, it's difficult to use a traditional 3D printing approach like fused deposition modeling to print soft tissues and organs within the body. For instance, if we try to print something like collagen in air, it just kind of collapses under its own weight. For example, this is a video where we're attempting to print a cube, but instead of end up with a sort of rounded hydrogel puddle. This is further exemplified in the images on the left, where you can see that as the number of layers of your soft material increases, you get this kind of deformation and collapse of the structure. And this overall goes to show that really when we print these soft materials, fidelity is lost. And then it's difficult to make these tissues and organs with 3D bioprinting. So how do we go about leveraging the advantages of 3D printing's soft extracellular matrix materials or cells, where we don't really want to print something like a blob, like this jello structure here, but we would rather print something like a complex 3D architecture, for instance, like this vascular tree seen on the right. Is there a way to print these soft materials like cells and extracellular matrix and maintain high shape fidelity? So one approach to think about this would be, and there are many approaches, would be to instead of making a tissue material printable in air, can we make an environment that makes any material printable? An alternative approach would be to use a technique which was developed in our lab, termed freeform reversible embedding of suspended hydrogels or fresh. This is a 3D printing technique that allows for the printing of soft tissue-like materials, or what we term bioinks. Here, a bioink, which is a gelling fluid, is extruded into a sacrificial support bath, where the bioink is shown in green and the gelatin support bath is shown in yellow. This gelatin support bath has a unique property in that it behaves like a yield stress fluid. And this is something similar to how ketchup behaves, where you have to supply a sufficient force to get it to flow like a fluid. For instance, you may have to hit the side of a ketchup bottle to get it to flow to the bottom. This happens when our needle is actually traversing through the bath, causing the bath to yield, and for our fluid gelling bioink to be deposited. However, once this force is removed, the bath tends to act like a solid, which enables the extruded bioink to be held in place while gelation or cross-linking occurs. Finally, because the support bath is thermoreversible, we can melt the support bath to release our final 3D printed structure. For instance, this video shows the printing of an alginate hydrogel, which is actually shown in blue in the letters CMU, into a gelatin microparticle support bath, which is shown in yellow. And then you'll see here, we can actually just melt away the support bath to release our 3D printed structure. So if we look more closely at the support bath, what we see is that we actually have two different components on the microstructure. So we have these tiny sphere structures, which are our gelatin microparticles. This allows for that yield stress phenomenon that I was talking about, as well as the thermal reversibility. We also have a fluid phase between these gelatin microparticles that we can incorporate a wide range of cross-linking or gelation mechanisms into. For instance, if we intend to print type 1 collagen or decellularized matrices, we can incorporate a strong pH neutralizing buffer into the support bath. Or if we intend to print something like fibrinogen, we can incorporate thrombin into our support bath. Further, by using multiple extruders, we can print multiple bioinks into the same construct with these distinct cross-linking mechanisms. And this is shown in this picture on the right, where we have the frame of this material, which is shown in red, which is type 1 collagen. We have alginate, which is shown in blue, which has an ionic cross-linking mechanism. We have fibrin, which is shown in pink. And further, we have this methacrylated hyaluronic acid, which is shown in green, which is a photo cross-linkable material. So how do we get to building a full heart? So first, if we look at the heart, we need to be able to build patient-specific volumetric scaffolds with precise anatomical architectures. We also need to be able to build heart valves that can control blood flow through the heart, as well as the blood vessel components that deliver nutrients and gas to keep this heart muscle tissue alive. And finally, we need to be able to build the dense heart muscle or myocardium, which consists of these heart muscle cells that are actually providing the contractile component or pumping capacity of the heart. And with Fresh, we really have been able to 3D print many of these components. For instance, we can take whole models of the heart. This is a patient-specific model that was derived from MRI. We can scale them down to the size of about a neonatal heart, and then print these structures entirely out of type 1 collagen, and even see when we look at a cross-section of this print that we really have these defined architectures. For instance, we have these chambers, and we have even these trabeculae-like structures, which are within the chambers. You should be able to appreciate the detail created in these prints from these micro CT images, where we can actually see the right ventricle here, the left ventricle with its trabeculae, and another component of 3D prints, which is actually this 3D printed rectangular infill, which you could imagine that we could incorporate heart muscle cells or other cellularized components into. Further, you can see that we can actually recreate these fine valve structures, such as the aortic valve leaflets, as well as the pulmonary valve leaflets. We can also use this to build whole heart mimics that are actually the size of an adult human heart. So this is the same heart model that's actually printed to scale. This is printed with alginate, and you can see that it's incredibly handleable. And the goal here really is to develop modules for pre-surgical planning that actually mimic the modulus of heart muscle tissue within our body. Not only have we been able to build these structural components that I've been talking about, we've also been able to build the functional components of the heart, like valves, which, as I said previously, allow for unidirectional fluid flow through the heart. For instance, this is a computer-aided design model of a Trileaflet heart valve, and this is that same heart valve printed out of type 1 collagen. And similarly, you can see that when we fix it with glutaraldehyde, it is very, very handleable. And you'll see here in a second that we can actually very easily separate the leaflets, which shows you what high fidelity we can actually achieve with this printing process. But perhaps what's more impressive is that we can actually put them under pulsatile flow. So in this video, you're going to see a cross-section of the heart valve within a pulsatile perfusion system. And what's very impressive is that you can actually see that these leaflets will open and close under pulsatile flow. And these were under pressures that were observed on the right side of the heart, which is very impressive. You can see here that we actually had failure of this heart valve after several cycles, but we believe with optimization of the printing process and printing parameters and even the model, we can definitely get these heart valves to survive longer. We are also able to computationally generate vessel networks to be able to provide nutrients to developing heart muscle. So here is a print where we actually have the coronary artery here. And what you can see is this dense branching vasculature off the coronary artery, which we've computationally generated, since typically these vessels are not seen on an MRI, and we need to be able to provide nutrients to our heart muscle tissue. We can actually then print this structure entirely out of type 1 collagen, as you can see here on the right, and then even perfuse it with a blood mimic, as you can see here in the video on the right. So far, I've been talking about generating these acellular structures of the heart and its associated components. But how do we actually incorporate contractile heart muscle cells that are responsible for the pumping capacity? And so what we can do here is take those induced pluripotent stem cells that I was talking about in the beginning of my talk, and we add some factors to them to mimic mesoderm induction and cardiac specification. And after a period of about 14 days, we get these spontaneously beating and contracting cardiomyocytes in a dish, as you can see in the video below. With this method, we have been able to generate simplified contractile pumps, for instance, like this heart tube model here. As you can see, so this is a heart tube suspended across two supports designed to hold the tube open. We can print this entirely out of type 1 collagen, and OCT imaging can be used to actually see that we have very high fidelity of this lumen on the cross section and as well as the longitudinal section. We can then generate a contractile cardiac muscle tissue around this tube. And after a period of about 14 days, we can actually see that these heart tubes are able to contract on their own and generate a propagation wave along the tube's longitudinal axis. We can also evaluate the ability of these tubes to actually pump fluid, where we can inject these fluorescent beads into the tube lumen. And if we actually zoom into the area just outside the tube lumen, what we see is that these tubes are able to contract and generate pressure within their lumens to displace these fluorescent beads away from the lumen. However, because we don't have any valve-like structures incorporated at this moment in time, we do tend to get backflow of the beads towards the lumen during heart muscle tissue relaxation. However, despite this, we do see this minimal unidirectional displacement of these beads away from the tube lumen, which is represented by these vectors shown here, with the green beads moving out of the tube lumen, compared to these red beads, which are embedded within the wall and show no net unidirectional displacement. We can also 3D print these cells and ECM components directly to create simplified ventricle structures. So here's one example of this, where you can see that we have collagen walls that are surrounding this ventricular muscle wall. And you can actually see, here is a picture of this actual ventricle model. If we look at immunohistochemical staining of these ventricles, we see that we have a nice actin organization between the two pink collagen walls, which is second harmonic imaging. And what's also very interesting is that we can use the actual extrusion through the needle and the print pathing to sort of direct this cardiomyocyte alignment. What's notable here is that you can see that we do tend to get an orientation of these striated cardiomyocytes along the print path direction, which is important if we're trying to engineer something that will have directional contractile force or pressure generation. Finally, not only can we print these cells, but we can also show that they are functional. So in this video, we can see, which will start in a second. Basically, the calcium wave flowing through the cells, which drives this heart muscle contraction that you've been seeing. We can also see that we can field stimulate these cells with platinum electrodes, causing them to contract all at once at both 1 Hertz and 2 Hertz field stimulation. And here's also a top-down view where you can see the spontaneous as well as the field stimulated pace contractions, where I hope you can appreciate that this chamber is actually becoming a bit smaller with contractions, in some ways how an actual ventricle would function. So building on these previous foundations, how can we use this to extend our capabilities with FRESH? So far, I've talked about building these simplified heart muscle structures like tubes or ventricles, but how do we begin to think about building something that's actually more complex or reminiscent of this myofiber alignment that's actually seen in adult heart muscle? So this alignment is actually evident in other muscle structures. So we chose to focus on skeletal muscle for this work, where you have these different architectures including these parallel or aligned muscle fibers in one direction, these unipennate, which have like about a 45-degree alignment, and these bipennates, which have 45 degrees on one side and negative 45 degrees on the other side. We can actually very precisely generate slice models for print pathing of these different architectures, and then print them entirely out of type 1 collagen on the bottom here. And what's probably more important is that when we incorporate cells into these scaffolds, what we see is that we have, which the cells are shown in pink, is that we have this alignment of myosin heavy chain, which is shown in pink, to the actual 3D printed filaments. And it's probably most stark in this bipennate structure, where you can see that we have different alignments within the same structure. And I do want to say that this work is unpublished, and was done by Maria Stang and Andrew Lee within our laboratory. We can also build bioreactors to create a multiscale vasculature to perfuse these tissues. So this is actually a video showing bioreactor assembly with this tube structure within the bioreactor. So here, the bioreactor is being assembled. And here is our tube structure in the center. For instance, this one has 2 tubes on either end. And here is an actual print where you can see that one side is being perfused with a red dye, and the other side is being perfused with a blue dye, which really shows the versatility of this system. We can also try, though it's difficult, we can also try to generate the capillaries, which are actually below the resolution of our current bioprinting system, where you can see here, we're able to develop these, what we call proangiogenic or vasculogenic bioinks, where we can actually get these small capillary like structures that are assembling on their own. Finally, we can also generate these patient-specific models. And for this, we've really been focusing on creating patient-specific wound patches using decellularized ECM bioinks and printing very large volumes of material. Here, we can actually get a 3D image of a wound defect, a wound bed, and actually generate a personalized wound patch to that defect, which is shown here with the 3D slicing actually shown in this image on the bottom left. These can also be actually directly implanted on the wounds with the idea that they may be able to help heal wounds faster. And this work is being done by Annie Berry, as well as Josh Tashman and Dan Chaworski within our laboratory. As I previously mentioned, an important part for clinical translation is really developing processing and monitoring, as well as validation, that we're able to print exactly what we're saying we're able to print, especially if this is to be used for a patient-specific application. So here, Josh Tashman and Dan Chaworski within our lab have actually generated this combined 3D printing system with an optical coherence tomography scanner. And they actually developed this cleared support bath, which you actually can see the print through. And then they're able to image directly into the print bath. Here, they were able to print some pretty amazing things, including this surface rendering of this kidney, which is shown here. And you can appreciate the fine detail through the internal part of the kidney. So really, in summary, what are we trying to do here? So Fresh really enables the printing of various components of the human heart and other tissues. Our next steps are really to build anatomically large, functional heart muscle pumps. And we would really like to be able to translate these to large animal and preclinical models and develop commercial applications via biomanufactured devices and tissues. Finally, I do want to thank everyone in my lab that has contributed to this work described in this presentation, as well as our funding sources described under here. And I would be happy to answer any questions. Thank you very much.